picture a bright yellow, barely scaly mushroom, one that really prefers the damp wood of alders. It carries this fiercely bitter taste in the wild, plus, measurable antioxidant activity hidden deep in its mycelium. Welcome to this deep dive. Today, our mission is to explore the ecology and, well, the very specific identification nuances of Pholiota alnicola.
Yeah, it's a fungus defined entirely by its contradictions, really.
Right. And we are pulling from field guides, forestry research, and phytochemical studies to understand exactly how it operates.
It's definitely not famous for being a culinary delicacy or, a deadly poison. Its entire story is about this incredibly specific ecological niche.
And some surprising potential in the laboratory, too. But the contradictions start right away with its visual profile. I mean, it's classified as a Scalycap, but it's remarkably smooth.
Yeah, ironically smooth for a Scalycap.
Exactly. So it's kind of like the black sheep of the Scalycap family. If you find it, you'll see a greasy, bright yellow cap.
Right, and that grease is actually a highly specialized layer of gelatinized cells protecting it from the damp environment.
But because it lacks those signature scales, it's super tricky to identify. I mean, it just looks like so many other yellow wood-rotting mushrooms out there.
Oh, definitely.
Plus, I was looking at the microscopic data, and it even lacks Pleurocystidia.
Which is highly unusual for its genus.
Yeah.
Pleurocystidia are, well, they're essentially microscopic guard cells found along the gills of a lot of mushrooms.
Right. The ones used to secrete chemicals.
Exactly, or to maintain structural spacing so spores can drop. But Pholiota alnicola just doesn't have them at all.
So without those structural guard cells and, without the scales, how is anyone definitively identifying this in the field? Is it just trial and error?
It generally comes down to a taste test, though. Not an enjoyable one.
Oh, no.
Yeah. If you nibble a tiny piece of the flesh, you're immediately hit with this intensely bitter flavor. And that bitterness is its primary defense against fungivores.
You mean insects and animals foraging in the forest?
Right, exactly.
But wait, is it hiding a lethal toxin behind that bitterness? Like, is it actually dangerous?
Actually, no. It completely lacks dramatic, highly toxic secondary metabolites. So things like the complex chemical compounds like amatoxins that cause fatal organ failure.
Like you'd find in a death cap.
Right. The Alder Scalycap doesn't want to kill you. It just uses that awful taste to convince you, or, a hungry slug, to spit it out and look for dinner elsewhere.
It's an elegant, low-energy defense, I guess. That makes sense. Let's look at where it's actually deploying this defense. I notice in the forestry data that this mushroom thrives on wetland hardwoods, especially alders, though occasionally willow or birch.
Yeah, that's its prime habitat.
But it's experiencing a massive population boom right now. So are they suddenly taking over riparian ecosystems?
Well, they are booming, but they're reacting to a broader tragedy in the forest. There is a severe ecological crisis happening with alder trees right now.
Right, the alder dieback disease.
Exactly. A pathogen called Phytophthora alni is ravaging riverbank habitats, killing off trees at an alarming rate.
Wait, so is this mushroom essentially an ambulance chaser of the forest, just opportunistically capitalizing on the tragedy of the alders?
Ambulance chaser is close, but it's more like a biorecycling crew arriving at a demolition site.
Oh, interesting.
Yeah, the Alder Scalycap is a saprobe, so its ecological job is to digest dead and decaying organic matter. It's booming precisely because there is suddenly this massive surplus of dead alder wood.
Wood that desperately needs to be broken down, right, to keep the ecosystem functioning.
Exactly. It's stepping up to clean up the mess.
But what really caught my eye in the phytochemical studies is how this fungus behaves when you take it out of that dying forest and put it in a sterile lab.
Right. When scientists take the mycelium, the root-like vegetative network of the fungus, and grow it submerged in a liquid culture, the organism completely changes its behavior.
It stops producing those bitter defensive compounds entirely, doesn't it?
It does, completely.
Just because it doesn't need to fend off insects in a laboratory.
Basically, yeah. Without the environmental stress of predators, it redirects its energy. So instead of bitterness, the submerged mycelium yields significant antioxidant activity.
Wow, really?
Yeah, over 20% in some tests. But the timing is really critical. It doesn't happen immediately. The studies note that the antioxidant activity specifically peaks late in the cultivation cycle.
Around days 21 to 28, I think.
Exactly.
But why does it take almost a month to peak? What's triggering that?
Well, it's a metabolic switch. For the first few weeks, the mycelium focuses entirely on rapid growth, just consuming the readily available liquid nutrients.
Okay, so it's gorging itself first.
Right. And once those primary nutrients start running low around week 3, the fungus switches to a secondary metabolic phase. It produces those antioxidants as a survival response to the dwindling food supply.
So to summarize for everyone listening, Pholiota alnicola remains this quietly interesting specialist of damp, temperate woods.
Yes, beautifully said.
It's not really a culinary prize, but rather an ecological indicator of riverbank health, and clearly a surprisingly potent biotechnological tool.
It really is a brilliant example of a species deeply synchronized with its environment. And, there's one final detail about its life cycle that illustrates that perfectly.
Oh,
Yeah, its spores actually require freezing winter temperatures just to break their dormancy.
wait, so they need the deep freeze before they can even germinate?
They do. It's an evolutionary mechanism to ensure they don't germinate during like a deceptively warm autumn week, only to freeze to death a few days later.
Oh, wow, that is smart.
Yeah, they wait for the deep freeze to pass, perfectly timing their awakening to colonize that damp wood right as spring arrives.
It makes you wonder, though, as environments shift and those deep winter freezes become less reliable, how does an organism completely dependent on the cold adapt its internal clock?
That is the big question.
Will those spores still wake up to do their vital recycling work? Definitely something for you to mull over next time you spot a bright yellow cap on a damp riverbank.